So there are collections of people who've made it to the ripe old age of 100 and more. Aren't they a beautiful cohort to look at the NRF2 induced genes in? And I would be surprised if this hasn't been done already. We've talked about it for years, and I know we've talked about doing it in animals, but doing precisely as you suggest. Here's a cohort of very old people who are quite healthy. Do they have some? Everybody wants to know, is there something magic about that? But are the NRF2 inducible cytoprotective genes part of that equation? And we suspect that they might be. The prolonged lifespan experiments would pretty much have to be done in mice. And again, there's someone at there's a colleague that I know we've talked to at the Institute of Aging who was interested in doing that. I'm not sure if that ever happened. I had a high school student who wanted to do a project here a number of years ago, and we got her working on nematodes, Caenorhabditis, which is a common experimental vehicle for those interested in looking at lifespan, because its lifespan is so short and it cycles quickly.
I did a lot of work on C. elegans when I was before I went to grad school, lifespan studies.
Well, we ought to get together and do this experiment again if it hasn't been done. One of the problems with sulforaphane research nowadays is I can't keep up with it all. No one can really keep up with it all, because there are 2 or 3 papers a week coming out on sulforaphane, and quite a few more on the NRF2 pathway. I mean, if you try to be a Renaissance person and know all the literature in this field, it's going to smoke you, because there are thousands. There are, I think, a couple of thousands of papers a year on NRF2. So it's very difficult to keep track of them all, and it's possible that someone has done those lifespan experiments with C. elegans. Again, we started. We didn't have much muscle in it. We had a high school student who then had to go back to.
I didn't see anything in the literature, by the way. I looked for C. elegans. I didn't see anything with sulforaphane that was published.
No, we don't have any preliminary data from that student. We also had, I'm looking on my shelf for the binder, it may not still be here. We had a colleague who was a bona fide expert on C. elegans who was going to do some studies, and I'm not sure that ever happened or if those were ever published. So, yeah, sorry. I wish I could jump up and down.
But do you agree with me here? I mean, knowing what we know about how sulforaphane is, as you mentioned, the most potent dietary that we know of activator of the NRF2 system, and knowing what we know, we do know that NRF2 does play a role in delaying aging, in brain aging, in tissue aging. It's literally lowering inflammation and reducing oxidative stress. These things cause aging.
Right. Many people seem to think that by understanding a bit more about how we can reverse this phenotype in people with acute aging, that this may also be applicable to normal aging and to perhaps slowing down normal aging. It turns out that all of us have some small amount of progerin, this protein, in our systems, and I believe the evidence is that it increases somewhat as we age, not certainly to the acute levels that you find in kids with progeria. So we're using sulforaphane to explore this. Since we got our grant, there's a very interesting and I think quite important publication from Tom Mistelli's group at the NIH showing that in fact NRF2 is very intimately related to this process in progeria, and that what happens is that progerin, the sticky protein that I told you is on the inside of the nucleus, actually binds NRF2 as it enters the nucleus to do its signaling thing, and it gloms it up against the inside of the nucleus, so to speak, so that perhaps among the chores of sulforaphane, or among the functions of sulforaphane, are to increase the amount of NRF2 that's coming into the nucleus, certainly, but it may also enhance clearance of mutant proteins and even progerin from the nucleus. So there's a huge amount that we don't know there, but it's an exciting approach to studying aging.
Yeah, it sounds like even just the mutant progerin seems like it's stopping NRF2 from being activated, and that
It's sort of
essentially
holding it
it's
up at the
stopping it,
gate.
right?
Yeah,
And
yeah,
so
yeah.
that's possibly part of the pro-aging effect as well.
Yeah,
So,
so.
yeah, I was reading somewhere, maybe you can confirm this, that so sulforaphane, does increase the activity of NRF2. Like NRF2, I think it's something like 60%. Like NRF2, is activated every 180 minutes or something like that, and sulforaphane bumps that up, a 60% increase in it being activated. So I don't know if that's...
Yeah, I can't comment on that.
No,
Sorry,
okay.
but I mean...
But it's activating it more often for sure.
Correct, correct. Yeah.
So that's, for me, what I want. Another question I wanted to ask you.
It upregulates its production. So if you look at RNA levels for NRF2 or for KEAP1, it's tether protein that's present in the cytoplasm. After treatment with sulforaphane, you see those levels going up. So 60%.
I think that was a translocation in the nucleus.
Yeah,
that was the increase that it was doing.
Okay,
so.
okay. Yeah, I unfortunately I can't comment on that. Just don't know. But,
yeah, I mean, that's okay.
But
I talked about it in
I'm sure it's true. I believe you.